Hello and welcome. Today we are talking with Dr. Bob White. Dr. White is the Dean of the College of Biosciences here at KCU, and he came to KCU in 2010 after serving at Children's Mercy Hospital for 17 years. During his time there, he directed his own research lab. He ran the DNA diagnostic lab, as well as being the head of the IRB at Children's Mercy. Prior to that, he was a postdoc at Children's Hospital with Harvard Medical School in Boston, and he received his PhD from Boston College, along with an interaction with the Jackson Lab in Bar Harbor, Maine. So, hello and welcome, Dr. White.
Hello.
So, why don't you go ahead and give us a kind of a 30-second or a short elevator pitch of the current research focus in your lab?
So we have 3 projects in my lab, and the focus is on finding new therapeutic strategies to treat some really difficult diseases. One is iron overloading. The patient compliance to treat iron overloading is a major problem, and we have, using a mouse model, found a new way to treat those patients. Secondly, we are using a mouse model to study a new way to treat a type of leukemia called AMKL. And then finally, as a mouse geneticist, we have been working on gene therapy for Duchenne muscular dystrophy.
Great. So it sounds like a lot of your work is being done in mouse models. Is that, would you say that's right?
That is correct. In fact, some of these mouse models we obtained from Japan, obtained from England, and we are the only ones in this country that has these mouse strains.
Okay. And maybe tell me just a little bit more about your muscular dystrophy mouse model. Is that like a knockout type of mouse, or?
Actually, it's a transgenic mouse, and it's a very interesting story because it began with an unusual finding in the ophthalmology clinic at Children's Mercy Hospital, which led to our discovery of human retinal dystrophin. So this is about 2/3 the size of muscle dystrophin. It contains the 4 functional domains that you see in muscle dystrophin. It's in the retinal dystrophin. When we discovered this, I wondered if the expression of this retinal protein in muscle would replace the lack of full-length muscle dystrophin. We went ahead and we established a transgene using a muscle promoter with Stephen Hauschka from the University of Washington in Seattle. And we were able to get this expressed in mice that have severe DMD. They develop scoliosis, they die in an early age, they have progressive muscle disease, and this all was prevented by the expression of retinal dystrophin in the muscle of the mice. So now what we're working on is how to go from the lab bench to the bedside of patients. And interestingly, it turns out that 30% of patients with Duchenne muscular dystrophy have the ability to make retinal dystrophin, and they don't make it in their muscle where it's not normally expressed. It's muscle, it's not retina. But what we're doing now is trying to identify the promoter for human retinal dystrophin. So perhaps we can find a drug that will induce the activity of the promoter to get that protein expressed in the muscle of patients. And hopefully we have the same outcomes that we do with our mice.
Wow, that's really interesting. So are you doing any work in human cells or human models at all?
Not at this stage. Well, actually we are. I'm sorry. We actually, in identifying the promoter, we need to have a cell line where that promoter will be active. And there is a retinoblastoma cell line that we have identified that does express retinal dystrophin. So when we're going through a bioinformatics process to identify the promoter, we will use that human cell line in order to identify it.
Wow, that's really interesting work. So my next question really kind of ties into that, and you can expand on your other projects as well, but maybe just mention why you feel your research is important and what some of the possible applications are for physicians.
So clearly, when it comes to Duchenne muscular dystrophy, it's a very serious disease. Patients die in their 20s, and a lot of very good work being done to try to find therapies for those patients. Our unique discovery of seeing that there's another isoform of dystrophin expressed in the retina has led to this, and the hope is that this may someday make a difference in patients. Don't know if it will, but we'll keep working until we know. When it comes to the study of iron overloading, it's very difficult to treat those patients. Typically, when you're identified with iron overloading from a genetic disease called hereditary hemochromatosis, you typically have to go to your doctor's office once a week for 2 years to have a unit of blood taken. So phlebotomy is the treatment of choice. And in addition, there are iron chelating drugs, which they use for these patients. They're slow in their action. It takes about a year to get the patients under control, and they have horrible side effects. Patient compliance is the biggest problem with treating patients with iron overloading and hemochromatosis. So we actually have another mouse mutant which excretes 100 times more iron in its urine than normal mice. It's not because there's red cells in the urine. It's not because of hemolysis. This is non-heme iron in the urine. We have actually mated our mice with hemochromatosis model mice and prevented iron overloading. So again, this is another case where if we can take this from the lab bench to the bedside of patients, it will definitely be translational. We have identified the genetic defect in our mice and the protein affected, and inhibiting the protein activity or decreasing the expression of the protein could lead to urinary iron excretion in these patients and perhaps better compliance. And then the study on the AMKL, acute megakaryoblast leukemia, was generated by studying a mouse mutant, which we thought initially was just going to generate some interesting basic science hematology. But what we discovered was this mouse has a mutation that leads to this leukemia in patients with Down syndrome. These mice produce a protein which is called GATA1 short, which is associated with AMKL and Down syndrome patients. But in our mice, they have the same kind of mutation that occurs in humans, but they stop making GATA1 short. They recover from their anemia. So clearly, if we can figure out the mechanism by which the mice are prevented from developing AMKL, we could have something that could be a pharmaceutical target for treating these patients with this leukemia.
Great. Wow. So you've got definitely 3 projects that really have a medical application almost directly. That's great. So how do you feel, or what do you think about working in your lab helps to prepare medical students for a career in medicine?
Well, if a student is interested in being a physician scientist, an academic physician, I think experience with experimental design and studies, careful data taking and analysis will help them greatly in moving towards those residencies that have a research component. I did have a student in my lab. He was a medical student in his 4th year. He spent 2 rotations in my lab, back to back, so it was 2 months full-time in my lab. And he worked on this muscular dystrophy project that I talked about. When he went for an interview for residency at a Mayo Clinic, which would involve research, and when they were interviewing him and they asked him about whether he had any research experience, he truly knew what was going on in the lab, and he had a story to tell. And that's all they talked about during the interview at that stage, once they learned that he had done research. And he did get the residency at this Mayo Clinic. And now as a physician scientist, spending part of his time in the clinic, seeing patients, and part of the time with those unusual findings that occur in the clinic, taking them to the lab to try to find new ways to combat disease.
Excellent. So why do you feel research in general then is important in medical school education?
Well, the way I look at physicians is that they're like the infantry down in the field fighting disease. They're the front line individuals who are fighting disease. When it comes to researchers, we're like, the intelligence group back at the home country trying to find new ways to combat disease. So, hopefully we come up with new ways to treat things like iron overloading, leukemia, Duchenne muscular dystrophy. And so I think that's where the value lies.
Great. So how many students or residents or fellows have you had the opportunity to work with and train in the past? And then what are some things that some of them have gone on to do?
Well, I've been blessed to be active in science and research labs for 28 years, and there's many students whose lives I have been able to touch, undergraduates, medical students from the UMKC School of Medicine when I was at Children's Mercy, and of course at KCU, the graduate students and so on. there's been so many. And again, a lot of them have gone on to careers where research was a component, and the experience of being in the lab, not only the techniques, but just, learning how to set up experimental design, how to ask questions, how to figure things out, I think has made quite a difference in their careers.
Yeah, I've been having a lot of discussions lately about the idea of physicians and their knowledge of what the scientific method is, and how to communicate that, and how important that can be. We see it directly with COVID. um,
Yeah,
you know
exactly.
uh, there's a lot of information and a lot of misinformation, and it's important that our physicians are able to understand what it is that's coming out of the labs, and be able to report that, and talk about it, to their patients in the clinic or in the hospital, um,
Exactly.
or just to the public in general, to get some scientific buy-in, whether it's about COVID or anything really.
Mhm.
So I'll kind of wrap it up here with one piece of advice that you would give to students, whether they're thinking about a career in research or a career as a physician scientist. What's one piece of advice that you would give to them?
Well, it's something that I tell every student who comes into my lab. Job number one is to understand the research so you can tell a story. You've got to be able to tell the story. So when someone asks you, so you did research, what did you do in the lab? Oh, I did some PCR reactions, I did some DNA extractions. I don't know why, but yeah, I did that. No, that's not what you need to say. You need to tell the story. You need to have the background and understand the work. So job number one is to understand the research so you can relay it to others. And I think that's one of the most important things.
Great. Well, thank you, Dr. White. I appreciate you taking some time to talk to me today about your research and what you do in your lab. I know the university appreciates all the students, and the research that comes out of your lab and the opportunities that you provide for our students. So it's great talking with you, and hopefully we'll have a chance to do this again soon.
Thank you, Dr. Kramer.
All right. You have a good day.